Hello, everyone. Welcome to our seventh lecture of 443 computational biology modeling methods, virtual tissues. Just as a reminder that the class is being live streamed on YouTube. We'll be talking today about diffusion, decay, and we will add secretion and uptake from cells. And we'll see how this type of processes can model actual biological processes in real tissues. We're gonna, as always, we're gonna start with a short student project updates. Remember that the idea of having this updates is for all of us to keep track of the progress. We don't want to run into last minute type of trying to come up with a model and uploading it to NanoHub. It is a process that takes time to polish and develop the models, to turn them into workable code that can be uploaded to NanoHub. So we want to have buffer time such that students can present their projects and we can receive feedback and then upload them correctly into NanoHub. Last time we talked about how to modify the growth rate of cells and the growth rate and division rate of cells according to pressure. And we talked about how this was intended to model actual biological processes in which cells communicate between each other and inhibit growth due to, for example, contact between cells. And this is a mechanism that cells rely on to enforce the right shaping of tissues, so that tissues don't overgrowth and so on. And this is also one of the mechanisms that in cancer cells need to overcome to be able to proliferate even in contact with other cells. We talked about how to think about molecules in tissues and how to represent them as and why and how to represent them as concentration fields. And we talked about what are the actual biophysical processes that lead to changes in concentrations across biological domains. We talked about how diffusion can be used to model the transport of molecules or the rate of change of concentrations, and under the conditions in which that model was a reasonable model of transport. And we talked about the importance of specifying initial and boundary conditions and how with the diffusion equation and with certain boundary conditions, we can achieve a gradient that it's linear across the tissue. And we'll come back to this again just to make it more clear this time. Then in this class, we're gonna talk about how we can represent membranes in CompuCell as regions of the domain that have different diffusion coefficients, and we'll show how to do that. We will talk about adding decay terms to the diffusion equation and how the concentration profile changes when decay is taken into consideration, and what these decay terms are meant to represent from a biological perspective. And then we will talk about cells as sources and sinks, or as areas of the simulation domain where these fields can be produced and can be uptaken, and how this secretion, what we call cellular secretion and cellular uptake or absorption, leads to changes in concentration of what we will start thinking of as the extracellular environment. Hopefully we'll get to a point where we can start simulating very basic behavior of tumors where we have different types of cells inside a tumor that might be that are dependent on how much exposure they get to metabolic resources. But before delving into that, let's just go around the room to get our usual project update. I'm gonna start with Devin.
Yeah, so my computer's still rebooting, so I'll just talk about what I've done so far. So basically, I've just initialized all the cell types and added all the basic plugins like adhesion, volume, stuff like that. And then I've also implemented the Diffusion Solver, kind of based on what we talked about last class. And then for this next week, I want to add the rest of the Python algorithms that are a part of the code, and then maybe start to implement some new expansions on the project.
Mm-hmm.
And that's where I'm at now.
Okay. Just as a reminder, we did not have weekly homework this week, but we're still expecting students to keep track of what their progress is in terms of the milestones that you've set yourself for in terms of progress in the project. So let's go ahead with Drew.
Okay.
Also, if you guys want to share screen, just let me know and I'll pass on the screen to you guys. Yeah, go ahead, Drew. Although it's not needed.
I'll just describe mine. It's not really much to show at the moment, but my goal for this week was to I've got two different drug delivery things for tumor cell simulations, and I wanted to get at least one of them set up with the basic information that I could get from the paper. So what I did know was like the size of the simulation and how many cells there would be, like how big of a blob it is, and I set that up. That was easy enough, but I also wanted to see about implementing diffusion stuff, and I was hoping to get it working within the past hour, but CompuCell's not really been working with me. But I'm going to try to finish that by today. But I was able to get some sort of like trackers down and other plots generated for my stuff, but I can't really show it to you at the moment because I can't. I'm having some graphical issues with mine. I don't know why.
That's totally okay. But just as a reminder as well, if you guys have questions that you come across anytime during the week, just send an email, and I'm pretty sure one of us will be available to get back to you and help you out. We've been doing it regularly with some of the people who have been contacting us. Fambo?
Can I share my screen?
Yeah.
So basically, in the last week, I've tried to figure out what is the DOM event, and the DOM event is related to the keyboard and mouse events that triggers in the web browser. And we found a very useful library called the IPY events. So I'm trying to go through all the information, the documentation inside IPY events, and try to do some basic implementation using the IPY events to listen to the mouse and try to deploy it inside an image widget. And I will show you the examples in the following. So basically, the DOM event is a short for the document object model. So it is basically a representation of the components of a web page displayed in the browser. So the DOM events corresponds to the keyboard and mouse event in the browser. So, and very simple example is the Jupyter Notebook seal. So if you interact with the seal, the key press and mouse clicks are generated by the browser, and it will handled by the notebook, by the JavaScript inside there. So a very useful library called IPRN, there's a very powerful library, and also it's a very new library which can listen to the mouse and keyboard events and wrap it with a custom widget. And this widget is called the event widget that can be set, that is very similar to like other traditional widget, for example, the image or label widget. But the only thing is the event widgets cannot be self-displayed, but it can be showed the information by other widgets, for example, the HTML or the image widget. So basically, if this is an example of how to use the IPR events use. So this is a label, and if you try to click on that, it will return it will capture like you click on that, you can see the type is click and based on the timestamp of that. And if you type on that, it will show you the type is key down and the code is the key A, S, Q, F, and W of that. So it will capture either the keyboard and the mouse enters of that. All the events are showing here is, I think it's very, very broad of that. It can capture all the kind of the mouse event and the keyboard event set. And another example is of our image widget. So if you see that, because we initialize our image widget to be 900 by 900, so if you click on that, it will return the instantaneous coordinates of the, if you found like your mouse locations to that, so that we can, and then we can use the function inside the Mechanica to rotate the screen and try to zoom in, zoom out the screen with the correct location. And if you click outside the image, it will not return anything of that. So there are two basic challenges I found that might be challenging the project. The first is because the IPR event is kind of an external thing, so it will require to add some notebook extension. So I don't know if you deploy it in the NanoHub application, it will also allow you to install such kind of extensions. And another thing is the mouse move event will triggers very often, like inside. So it will have a very large volume of events between the front end and back end. So that means it will gives you, it may cause some lags like between the front end and the back end. This is a task for this week, and I'll show you in the next week. So basically, I will implement 3 of the like very basic functions of that. Trying to rotate the camera, use the mouse, and trying to translate the camera, use the shift button, and also use the mouse. And also zoom in and zoom out, use the mouse wheel. Yeah, that's all. Thank you.
Andy, and yeah, just feel free to jump in if you want to comment about the student projects as well. Let me put back the and then Jeffrey.
I can share my screen.
Yep.
So first, I've been trying to calculate the pressure, and I ran into a few bugs. So I've been working around it, and these functions I've made so far don't work as I expected. But the one that I think will work, that's in some papers on dissipative particle dynamics, involves the stress tensor, and I think the bugs have been fixed, and I'll be able to add that after class and check that it works. And another issue was the particle density at the boundaries. You can see, I guess here, it's higher at the edges. And one solution to that is to just lower the density. See here, it's more or less straight. The scale is around, I guess, 0.3 difference here, and here it's 3. The boundary potentials is another solution, but I'm having a difficult time getting it to keep the flow profile. It separates it from the boundaries a little too much. So you see it's just a straight line moving through. And then with the driven cavity, I've gotten the streamline plots to be a little better. You can kind of make out the curls in this, and then the color underneath it is the magnitude of the velocity. So it's a little higher up here, like it should be, and lower, but it still could probably use some improvement so that I'd prefer these to be a little smoother. And next I have the chemical species, and we talked about putting a stationary object in the flow so it flows around it. So I think, yeah, that is all.
Yeah, that looks really cool. So yeah, I just I want to remind people that just to keep meeting with Andy, TJ, me, and James at least on a weekly basis, so we can help you also keep track of your progress. Tyler.
Yeah, I have a few updates. So my goals for this first milestone were to implement my 5 different cell types, including the contact energies, the volume constraint, etc. And I wanted to start implementing the intracellular ODE model in my project. And if I could, start working on cell differentiation, which I didn't really get around to this week. But when I run my simulation, I've come into a problem where my starting cell count is correct, but the volumes are not. So I'll work on getting that correct this week. And I would also like to finish implementing the ODE model so that cells differentiate according to their contacts and various signaling thresholds. And then I'd like to, if I can, start implementing the apoptosis. And that's about all I have.
Cool. Yeah, it seems like everyone is doing consistent progress. That's exactly what the purpose of this weekly assignments are. And again, I want to encourage everyone to reach out with whatever problems you're coming across. So coming back to the class, the last time we were reviewing what happens with cell division in CompuCell when you specify a constant cell growth. And we showed that in the case of constant cell growth, you reach a point where the cells fill up the simulation domain. Do you guys hear me well? Yep. Okay. you guys, so the cells reach, there's a point where the cells fill up the simulation domain, and as they fill the simulation domain and they run out of space to grow, the pressure inside each cell keeps increasing and blows up. And we talked about one way around that solution was to having one, at least to slow down the growth rate of cells, is which is something that you actually see in tissue, is to have one specialized cell type that grows and divide, and then another cell type that we call quiescent, that are usually there to perform specialized functions, and they are thermally differentiated or arrested in the cell cycle. We also talked about another way in which cells and actual real tissues regulate the pressure along the epithelial sheath is by having mechanisms that inhibit cell proliferation and division when cells come into contact with each other. And we talked about how this is one of the main mechanisms in wound healing or repairing. When your stained cells are actively proliferating around a wound, you will see that those cells that are at the edge of the wounds are usually the ones that are proliferating the faster, whereas the other ones deeper into, let's say, the intact tissue, they usually send signals and such, but they're usually not proliferating, and it's the availability of space for cells to grow into that drive this cell growth. And then once cells have, let's say, closed the wound, they stop proliferating, and this is driven in part by what is called contact inhibited growth, where cells communicate to each other that they have no more available space and they stop proliferating. And we talked about how one way of representing that in CompuCell was to modify our growth rate to make it a function of the pressure the cell experience. And by pressure, we mean the difference in target volume and target surface, target volume and actual volume. And I should point out here that I was talking to James about this earlier. We have a little bit of a convention mismatch, because what you can define the sign of the pressure whatever direction you want, right? But conventionally you would define the pressure. So for example, if the for example, if the target volume, if the actual volume is smaller than the target volume, the pressure is driving the cell to grow, and that pressure outward would be positive. Whereas if the target pressure is, if the actual volume, is bigger than the target volume, then the force is driving the cell to shrink, to go inside, and we will describe that pressure as being negative. It is a convention, but just in terms of how to conventionally describing it, it's better to adopt that convention. And so pressure would be defined in this, using this equation, right? Which is proportional to the difference between the target surface and the actual, I keep saying surface, the target volume minus the actual volume. And then, as I said before, one of probably one of the simplest ways of representing this contact inhibition is by making your growth rate a function of this pressure that cells is experiencing. And one of the classic equations to modulate processes or to regulate processes is to use this Hill equation or Hill function, where you experience some sort of activation or inhibition, right? Where as the pressure increases, these terms become smaller and smaller, and then you can multiply your maximum rate of growth by this term, such that as the pressure keeps increasing, then your growth rate goes to zero. Part of the homework will be to explore what happens when we change the parameters of this functional form. But again, the idea is, let me see, can I draw here? Yes. The idea is that if we were to plot the growth rate as a function of pressure, when the pressure is near zero, that growth rate will be at a maximum. And as the pressure increases, the growth rate decreases and goes to zero. And then that K parameter determines where the inflection point happens in this curve. As you increase this parameter, your growth rate function starts looking more and more like a step function. So if you wanted to model a process that looks a little bit more like a switch, where cells stop dividing once they experience a particular, once they surpass a particular threshold in pressure, you would increase this, what we call the Hill coefficient parameter here. Okay. And this is important because this is gonna be part of the homework, and this is what I was trying to show you last time, but I didn't get to run it all the way through. So as I make this parameter here, or as I vary K, I go from a simulation where the pressure increases slowly to a simulation where the pressure increases much and more faster after cells have reached that have fully filled the simulation space. We call that confluency in experimental biology. Okay, right. But the point of the whole, and you'll play with these parameters a little bit, and the idea in the homework again will be to compare the growth rate and the average pressure as you modify the parameters of this equation. So after talking about, spending a couple of weeks talking about cell growth and division, we added another level of complexity, another way of representing more and more biological objects and processes. And we talked about how to represent the presence of molecules in our tissue. We talked about how in the context of tissues, we can treat these molecules, because of their density, we can treat them as chemical fields instead of as concentration fields. Instead of treating each of the chemical molecules independently, we treat them as a concentration, right? As a number of molecules per unit volume. And then we can describe how these molecules behave in the tissue how they move across the tissue by describing and modeling how the concentration changes. And we talked about a couple of mechanisms that drive these concentration changes in biophysical systems. We talked about advection, and we talked about active transport. Active transport occurring, for example, at the interface of cell plasma membranes, where they have lipoproteins, right, that are responsible for taking stuff in and pushing stuff out of cells. That usually requires some energy. It is expensive to the cell in the sense that they have to use metabolic resources to carry out this active transports. But over short ranges, cells actually take advantage of another very important transport mechanism. Just one second. Cells take advantage of this other. Well, just one second. Cells take advantage of this process called diffusion, where just driven by the differences in concentrations between areas of a particular domain, molecules move from areas of high concentration to areas of low concentration driven by their molecular motions. And the reason this mechanism is very important in biology is because cells can take advantage of it without having to use metabolic resources. It arises spontaneously from molecular motion and differences in concentration. It's important to notice that diffusion is mostly important over short distances, usually a few millimeters, and becomes less and less efficient as we go over longer and longer distances. And we mentioned that, for example, if a neuron that is some neurons can actually be very long, that if they need to transport some neurotransmitter across the cell body, they would usually rely on some type of active transport rather than in diffusion, because of the efficiency of the efficiency of diffusion diminishes greatly with distance, and we'll talk a little bit about that too today. So we also talked about how in real tissues, diffusion is not uniform. It's modulated by the density of the medium they are diffusing through. They are modulated by encountering cell membranes that can be permeable, semi-permeable, or impermeable to these molecules. And all of this will change the rate of diffusion of the molecules across tissue. And one of our first exercises today will be to play a little bit with changing the diffusion coefficients in different areas of the simulation domain to represent cell membranes. We talked a little bit too about how to mathematically model diffusion. And we said that diffusion is driven by the change in flux or by the gradient as described in this equation. And that's why when we talked about when we initialize a simulation with a uniform concentration across the simulation domain, we do not get to see any changes in concentration because there's no gradient. And if there's no gradient, you'll see no diffusion. And this is important too when we want to calculate what the concentration is at equilibrium states. But we're not going to go through deriving the diffusion equation from the Fick's law, which describes the flux across, let's say, a particular area of a simulation, or a particular area or volume. But we will. But if you guys are interested, we're gonna upload this presentation, and at the end of the presentation, we've tagged reference material where the derivation of the diffusion equation is explained. What is important to know from here, right, is that the change in concentration in time is gonna depend on the gradient of the concentration, right, and how the gradient changes in space, times this diffusion coefficient, which sets the scale at which the molecule, or the time scale, how fast the molecule is diffusing across tissue. So, for example, we know that molecules that are bigger and heavier diffuse slower than molecules that are lighter and smaller. And we will represent that differences between molecules by having different diffusion coefficients as well. This is a way of modeling the differences in the type of molecules that we are representing, or the type of molecule the field is intended to represent. We talked about boundary conditions and how to fully characterize a diffusion within a system, we need to specify what happens at the edges of the system. And we said that there are three possible things that can happen to the field or the concentration at the boundary. One of them is that in the periodic boundaries, what the concentration is at one edge is basically reflected at the other edge. And if you want to think about it in this way, you can think of it as the simulation domain that we are representing being a representative sample of a bigger domain that is just our mirror image of the one that we're looking at, such that if something comes out on the left side of our simulation domain, it will come through the right side of our simulation domain. Other options for boundary conditions are constant flux, where there is a constant influx or outflux of material from the simulation domain, and constant value, where at the edges we set the concentration to be constant, invariant in time, and it's always the same concentration. And we play with this differences in boundary conditions a little bit. I will upload the demos, so if you want to give it a try on your own, it will be in the Canvas site. Lastly, we talked about how to represent this field, how does CompuSol represent these fields. We mentioned that CompuSol represents these fields as basically numbers that belong to each of the lattices of our simulation domain. And these numbers represent what the concentration of the field is at that particular location. So that's going to be important because that's what determines the direction where diffusion is going to happen. And it's going to be related to how much cells, how much of the material cells have access to, depending on their location. Right. So last time we end up class talking about, right, we practiced a couple of we had a couple of exercises with changing diffusion boundary and initial conditions and seeing what would happen on those cases. And we end up class, and I want to take a little bit of time to review this because this is important, talking about what happens when we simulate diffusion with no decay and fixed boundary values. And what we said that is in that case, right, in that case, at equilibrium, the change in concentration in time is gonna be 0. So at equilibrium, the value of the field at a particular location remains constant. And if that is the case, then we can solve the diffusion equation, right? So by integrating this term, and by integrating the gradient term here, and what we get is that the profile of the concentration is given by this equation. This is important because this is going to be in the homework as well. And then once we have this equation, we can plug in the values at the edges to get what the concentration is, what the actual form of, or what the values of this constant are in the concentration profile. And what is important to notice in this particular case is that the concentration increases or decreases linearly out of a particular concentration at location 0, which we call in here constant 2, right? So and that is important because what we expect to see is if we have a simulation where we have a fixed value at one edge and a fixed value at a different edge, and we have just diffusion between those two edges, what we should expect to see is a linear gradient between the edge, that acts as a source, right, and which is the one that has the highest concentration versus the one that has the smallest concentration. You'll see a linear gradient from one to the other one, and that linear gradient is described by this equation. Right. So this is exact. I should have moved forward because this is just explaining exactly what I was just talking about. It's important to notice that that you see that the concentration profile at equilibrium is independent of the D parameter of the diffusion coefficient. But how long it takes for the system to equilibrate, that will depend on D and how big the lattice, how big the distance between the two edges is. And when we implement that in CompuCell, what we get is that we start with a high concentration of the field at one of the edges, and as the simulation progresses, we see that the concentration, right, reaches a point at equilibrium, right, where it's not changing anymore, right? So this curve is not increasing, stays stationary, and becomes a straight line between the value at the source edge and the value at the sink edge, or the value at where the edge is smaller. And of course, the slope, the equation for this line is just what we saw in the previous slide, and that's going to be important too, because one of the exercises of the homework is to compare the theoretical equilibrium, right, versus whatever you have in the actual simulation, and see how close you are from, or how far away are you from that equilibrium concentration. Okay, any questions so far? Because I think next time, right, we're gonna, this was just a review of what we did last time, and we're gonna start delving into new material from this point on. Okay, I'm gonna stop sharing for a second so I can share my full screen. Okay. And this is what we wanted to talk about, and this is also why we wanted to explain a little bit why diffusion is very efficient over short distances, but very inefficient over long distances. And the reason is because the time that it takes a molecule to diffuse a distant L is gonna be proportional to the square of that distance. So if one cell takes, if a molecule takes, for example here, if the molecule takes 10 seconds to traverse a distance of 2 micrometers, it takes what did I say? I forgot, I'm sorry. I was just coming up with a number, but let's say it takes 2 seconds to traverse a distance of 2 micrometers, then it takes, well, because 2 by 2, but like, let's say it takes 16 seconds to traverse a distance of 4 micrometers. So as the cell keeps increasing, at the distance keeps increasing, the time that it takes the molecule to traverse distance, a certain distance is going to be proportional to the square of that distance. So, and there is an example here that if a typical protein has a diffusion coefficient of 100 micrometers per second, that molecule will take, to diffuse 1 meter, it will take 300 years. So of course, if you wanted a molecule to traverse your body from, let's say, your head to your bottom, just by diffusion, you'll have to wait 300 years to do so. So we have extracellular transportation systems like your blood vessels or your lymphatic system, but even through cells, we do have active transport mechanisms to try to transport molecules even over distances that are not necessarily like your whole body, but it would also take a decent amount of time to do so. Okay, so I want to come back to this idea that that in actual tissue, the diffusion coefficient, the diffusion of the molecule is not uniform and varies according to the density of the medium the molecule is diffusing through and whether they encounter barriers such as cell membranes or other physical barriers. And the way we will, one way of representing this varying in diffusion through tissues is to represent different areas of the simulation domain that have different diffusion coefficients. And what we'll do is that we're gonna create a simulation. We're gonna introduce a membrane between the edges of we're gonna go back to having a source and a sink at the boundaries, but we're gonna put a membrane in between and see how the membrane changes the diffusion that you would normally see throughout the simulation domain. Let's launch Compusol. I'm gonna ask everyone to launch Compusol. I might ask people to work through the code with me at some point. So I would ask everyone to have Compusol up and running, and then maybe we will. That's important. That's why it's important. James, I am actually not that good at generating the polls. So for this exercise, we're gonna need the polls. So I know everyone is up to date, and then I can, for example, for the second or third part, ask people
Sure.
to show
So
them.
if you go to the bottom of the screen where you have the
Yeah.
controls,
Right.
there should be a button next to participants called polls.
Mm-hmm.
Click on that.
Okay.
What
I
happened?
did. I have the I see the poll, and it's asking me
Right.
whether
So
to
that
launch
if
it.
you hit the blue button launch, then it will work.
Okay. And when does it stop?
So you watch until people have clicked on it, and when you're done, it will be end. There'll be a button called end polling.
Okay. Cool. Cool. Okay. So we're gonna start. I'm gonna work on this with you together. I'm gonna launch the poll now, just so people can let me know how they're doing through the exercise. So we're gonna launch to edit, and we are going to create a new project. And I already have a project called Membrane, because I did not want to run into the same problems as before. But what I'll do is I'll call it Membrane 1 to build, because it's not very complicated and we can build it together. Membrane 1. Right. So let's make the simulation domain not too big, so it doesn't take too long to solve the for the diffusion solver to solve the diffusion. And we're gonna need the rectangular slab. We don't care right now about what happens with cells at the boundaries, because cells won't be doing anything in this particular simulation. We hit next, and then let's create a cell type called membrane, and we're gonna freeze it. So cells are gonna be stationary. Let's add the oxygen field that we've been playing with, and we're gonna add it using the Diffusion Solver FE. And we don't need any of the sub-properties or behaviors, because as I mentioned, we're not gonna be using cells. They're just gonna be there so we can specify differences in the diffusion coefficient. Finish the simulation, and we come to the XML file. Okay. This is the part that I struggle always with a little bit zooming in and out. So we have our pods specifications. We're not gonna change that. We have our cell type membrane that is frozen. We know that it's frozen because it has this tag here in line 24 that's called freeze. And then we can come to the diffusion solver, and we see the field that we just created. It's called oxygen. At the moment, we are not dealing with decay. We're not including decay, or that we'll do later in class today, but we're not including it now. So we're gonna set that one to 0. And then we can see, we talked about this one last time. We talked about how this global diffusion constant specifies the diffusion of the field across the whole tissue. That's why it's a global diffusion constant. But now that we have added a cell type, we have these specifications that such that we can modify the diffusion coefficient by cell type, right? And what we will do is that for now, we're gonna set them to be the same as the global diffusion coefficient. We're gonna set them to be 1 and 0. As we continue going on the code, we see that the default snippet, code snippet, has this secretion data. The secretion data, and we will talk about that yesterday, specify how much of the chemical field the cells released to the simulation domain. For now, we don't need it because we don't want our cells to release any field, so we can delete or comment out that part of the code. And then, as I said, the boundary conditions will be, one edge will be constant value and it's gonna be 1, and the other edge in the X domain, it's gonna be constant value, but it's gonna be 0. So one is always gonna be 1 and always the other one is always gonna be 0. On the Y-axis, we can comment out the constant derivative, and what we need is periodic boundaries along the Y-axis. Okay, and then now we have set up everything that we need for the field, right? We have specified the diffusion coefficient. We've said that we don't want any decay, and we've also specified what the diffusion coefficient will be inside our cells. But for now, we've set those to be the same as the global, as in the rest of the tissue. But that would give us control later to change the diffusion of the field through the cells. And then what we want to do is basically we want to draw a membrane on the X domain that goes all the way up on the Y domain in the middle of the simulation. So what we will do is that we're in the X minimum, we know it's 100 by 100, let's do 45 to 55. And on the Y-axis, we want it all the way up. So we're gonna go from 0 to 100. And let's make the membrane width be 10. Okay. We save the simulation. Then we can come to CompuCell. Open our simulation. And launch it. So I just stepped it one step so we can see the layout of the simulation domain. So we have, in terms of our cells, we have created this frozen membrane here. Cells are, if I play it, you'll see that nothing is happening in terms of the cells move. And that's what we mean by frozen cells. And I'll launch it again. Oh, I did not want to. I wanted to. And instead, we're gonna look at the oxygen field. Wait, something's wrong in our model specification. No, that's fine. let's look at the configuration settings to make sure that we see one of the problems, and you will always have to be careful with this. Your configuration settings might be set up such that the colors displayed for the field are fixed or are floating. And those configurations will drastically change what you see in the simulation domain. Even though the underlying code and algorithm is solving the diffusion equation correctly, in terms of the visualization, you won't be able to see what's happening because you have the incorrect configuration settings. What's happening in this case is that I fixed the values at 0 and 10, but the maximum value I can get is always going to be 1 because that's the maximum value at the source. So I know that, since I know that the maximum value is going to be 1, I can set the visualization settings such that the highest color that I see or the highest concentration that I see is 1. Right, and we can see it there. And as we play the simulation, we see how the chemical is diffusing across the membrane, right? Because we set up the concentration, the diffusion coefficient of the field of the inside the cells to be the same as in the rest of the simulation domain. One way we can change this, I'll come back and show how it looks like if I change it dynamically in the model editor. But okay, let's see. There's 4 people who are with me in the 5 people who are with me. So maybe I can ask someone to share the screen now so we can play a little bit with this. Or I'll show you I'll show it first and then we'll come back with other exercises. With other changes and I'll have someone to show it. So let's do something. Right now we just model a completely permeable membrane, right? The membrane, the molecule is diffusing through the membrane as if the membrane was not there. But let's decrease the diffusion across the membrane. So the molecule diffuses 10 times lower across the membrane than it does across the tissue. I come back to the XML file, and here when I see diffusion coefficient by cell type, then I make it 10 times smaller than the global diffusion coefficient. And that means that the field is going to diffuse 10 times slower in the membrane than it does in the extracellular environment or in the rest of the simulation domain. And this way we will be simulating a semi-permeable membrane. Let's come here and let's play it again. So can someone describe what they're seeing now? What's happening?
The oxygen diffusing through the cell, and as it goes through the cell, it slows down. Right. It slows down because the diffusion coefficient, I specified it inside to be slower, right? That is determined by the diffusion coefficient. And then we can see that on one side of the membrane, the concentration is quite high, whereas in the other side of the membrane, it's still coming through, right? It's semi-permeable, but it's coming through. It's as we see by the ISO curves,
it's the concentration, it's going much slower in through the membrane that it's going outside of it, and such the concentration is smaller on the other side of the membrane, right? Okay. Anyone wants to take the lead now so we can change, we can make the membrane impermeable? I'll direct you. It's just a matter of you showing your code and doing it in your computer. If not, it's okay. I'll do it, but I want to give people the opportunity to do so if they wanted to. Okay, so I'll do it then. We've set the membrane to be semi-permeable in this case. If we want to make it impermeable, then we can make the diffusion inside those cells to be 0. So basically the molecule is not diffusing through that membrane. And what do you guys expect to happen before I run it? Any ideas? So if the membrane is impermeable, the molecule won't be able to get through it, and it will basically act as a barrier. So you have diffusion happening from this source here, but the molecule is unable to go through the membrane, and it gets stuck in between, right? It would act as what we've talked about as a no flux boundary, right? Where the molecule is diffusing this direction, but it's once it reaches the wall, it has nowhere to go. So at the end, the concentration of this side in this side, because of the constant value at the edge, the concentration in one side of the membrane equilibrates to be the concentration at the edge. Does that make sense? Okay, so let's do something fun. Let's simulate now a hole in the membrane. So just like cellular membranes do have pores through which some molecules can passively, I said actively, but can passively diffuse through, right? Due to differences in concentration between the inside of the outside of the cell. And that is particularly important when stuff is coming into the cell. And it's James, if you correct me, water is one of those molecules that diffuses passively through a lot of the cell membrane.
Yes, I mean your cells spend something on the order of 15 to 20% of their energy budget pumping water out of cells. And so as soon as the cell pumps are turned off, if your cell's ATP level is depleted, so the pumps aren't running, the cell will swell up and eventually burst. That's called hydrophilic swelling, and it's an indication that the mitochondria in the cell that produce the cell's energy are not working properly.
Right, and the reason that it happens is usually because the cytosol of the cell is usually very packed and very dense relative to the extracellular environment. So water diffuses in. What we'll do is that we will copy this uniform initializer snippet code. All right. Okay, and we're gonna paste it below. And now what we want is to make a hole on the Y domain, right? Because right now our Y domain goes from 0 to 100, but let's make it from 0 to 48, and then from 50 to 100. So in this case, we are basically creating a small opening in the same membrane, in the cell membrane. And let's look at how it looks like when we initialize the simulation domain. Right, that's a very small one. We actually play with bigger ones, but we can test this one, and then we can increase the size of the opening in the cell membrane. As we run the simulation, so we see that the chemical is actually going through the opening in the membrane, and it's starting to get through the across the cell membrane into the other part of let's say inside the cell, if you want to think about it in those terms. And it's taking a very, very long time for it to do it. The time that it takes for the chemical to cross the membrane is going to be somehow proportional, related to the width of that pore. So we are seeing that in this particular simulation, and to the diffusion coefficient as well, right? So I could come here, for example, and I could increase the diffusion coefficient. I could come to the, to the model editor, and in the diffusion data, I could increase the global diffusion coefficient. Let's make it 2. And that would make my field diffuse much faster across that membrane. So you guys see what I want to get is the ISO curve to come through, but they're going super, super slow. So what we're gonna do is we're gonna rerun the simulation, but with a bigger of a pore between them. Let's make it 45. Let's make it 46 to 64 to 54. My nanohub is trying to get a little bit slow. So we open up the and then we can actually see the ISO curve coming through and coming out from the other side of the membrane, and we can start seeing that it comes out. When it comes in, it starts to come in linear as uniformly distributed on the y-axis, but as it approaches the hole, it bends in, right? And then it comes out circularly on the other side. And I think a good exercise would be to measure how these isocurves are moving through the membrane, the pore, as a function of the pore diameter, if you want to think about it as some way. But you guys can see this way. And then if I increase the diffusion field now, the diffusion coefficient data, it should start diffusing much faster. So yeah, my nanohope is a little bit slow, but it looks much better in my computer. And it should look better on yours too. Okay. Any questions with this particular exercise?
Maybe just hit tile so that you have a bigger, you're occupying more of the screen, so it's easier to see.
Yeah, do you see me like coming like this close to the screen so I can actually see it? I'm sorry. And then you can see the isoforms. It's very sensitive. There. The isocurves as they come through the other side of the simulation, of the pore. Okay. Anything else you want to show, James, with this particular simulation?
Well, I think that one, I think it's useful for people to try that and play with it a little bit, because it gives you a sense of how, the other thing you could do,
yeah, just 1 second, because I forgot that I wanted to show that you could dynamically change. For example, this is the diffusion coefficient in the membrane, so I could dynamically change it. So now I made my membrane fully permeable again in the model editor, and let's make it semi-permeable. So I can dynamically change it as the simulation goes by.
that was actually what I was going to suggest you do.
Oh, I'm sorry.
Which is if you make the membrane only a tiny bit permeable, you can say how permeable does the membrane have to be, and how big does the hole have to be for the amount of chemical getting across the membrane to balance the amount through the hole.
Right. Again, if I after running the simulation, if I set it to 0, which I make it impermeable again, the amount that is through the membrane is gonna get trapped in it, because it's not diffusing anymore. So it basically got trapped inside the cell when I changed the diffusion coefficient inside the membrane dynamically.
It's also nice because it shows you how long or short a time it takes for the chemical field to equilibrate again.
Right. And we'll see that again when we play again with when we include decay. We can play with the diffusion coefficient to see how fast it goes back from the linear to the exponential. Cool. But yeah, there's this is a very interesting. I'm gonna upload this demo, but I do want to keep going so we cover other stuff that is also interesting. Let me end this poll. Okay. Yep. So far we've been talking about diffusion as if the molecule was stable across the simulation domain. And by that, I mean that there is no reason the molecule will disappear out of the simulation domain except whatever I set at the boundary conditions to happen, right? Except because of, let's say, I establish a flux out of the simulation domain that takes out the concentration, or if I set a sink by setting the value at an edge to be at a particular, like at a constant value, right? That's the way we've been playing it. But in real biological systems, molecules are not entirely stable across the whole tissue, because there is many mechanisms that interact with these molecules such that they are used up, or they decay or they are degraded for multiple reasons. Usually proteins are pretty stable in the sense that they do not spontaneously decay, such as radioactive material does, because yeah, their radioactive decays takes very, very long time. So when proteins and some of these biological molecules get degraded as they diffuse across the tissue, it's usually because some type of chemical reaction is happening that is leading to the degradation of these molecules. And we can include that into our model. So far, we have excluded all of that from our model of diffusion, but now we want to start including this decay. And what we will assume is that the rate at which these molecules are consumed, or the concentration decreases, is going to be proportional to the concentration that is already there. And that's what we call first order kinetics, first order decay kinetics. And that means that as the concentration decreases because of this first order decay kinetics, the concentration will never go negative, right? It's always going to be proportional to whatever is there, but it's also going to decay exponentially. The way the diffusion equation, the way the diffusion equation looks like when we introduce this decay term is, we are already familiar, is this way. We're already familiar with this term. This first term describes the change in concentration at a particular location x in time. That's what the diffusion equation is going to describe. D is the diffusion coefficient. We have explored already the effect of the diffusion coefficient, and the gradient, right? This is the diffusion term, but we are now adding this decay term that is going to be the rate at which the molecules decay and how fast the concentration decreases, because of this term is determined by this decay constant. For those of you who took the class last semester, this should start looking very familiar, because this was usually a dead term that we would include, for example, when we were modeling cell death or some population dynamics, for example, right? The death rate of the cells or individuals in the population is usually described using the same first order decay, the first order decay rate. So as I was mentioning, in living organisms, the molecules have what it's called a characteristic diffusion length, and that is determined by both how fast the molecule is diffusing, but also how fast the molecule is being used up, how fast the molecule decays in tissue. Right. So and this is going to be very, very important, this diffusion length, because this diffusion length sets what are the characteristic length scales at which certain processes happen. So for example, the diffusion length of oxygen out of a blood vessel determines the distance between two blood vessels such that, or capillaries, such that cells are properly oxygenated, right? Because oxygen is going to be used up by cells, so the distance between two capillaries, or how many cells can be between two capillaries, or before more oxygen has to be added, it's going to be determined by the diffusion length of oxygen. As oxygen, there's many important processes, especially in embryonic development, where signals are very important that determine the how fast the signals are go into the tissue, and the resulting patterns that arises because of the diffusion lengths of these signals. So I'm not gonna go over the whole math, but the idea here is, again, the same idea, right? That we want to know how the concentration changes in time at equilibrium, right? And by at equilibrium, we mean when, or we want to know what the right way of saying is, we want to know what the concentration profile is by solving the diffusion equation at equilibrium. And this is going to tell you how the concentration changes as a function of the location once there is no change in concentration in time. And this is just solving the equation, but what is important to notice is that when we solve the equation under those conditions, again, the condition is that we have decay, right? And that the concentration is at equilibrium, so it's not changing in time. What we notice is that the solution to that equation, it's no longer linear as we saw in the previous case, right? In the previous case, when we solve the diffusion equation at equilibrium and we set the boundary values, what we notice is that the concentration decreased linearly from one edge of the simulation to the other one. When we include decay, that concentration profile is not longer linear between the source and the sink, but instead decreases linearly. Decreases exponentially from the edge to the sink. And the diffusion length, what I was talking about, this characteristic length of the diffusing molecule is determined by the distance over which the molecule decreases a factor of E, right? What is the name of E in English? I don't remember, right? But by the factor of the exponential, right? And we can solve that right here. If we solve the equation by taking that into consideration, given the initial concentration, when is the concentration E, what we get is that that distance is determined by the square root of the diffusion coefficient over the decay rate. And that's what I meant by saying that the diffusion length increases as the diffusion coefficient increases, right? Because the molecule diffuses faster, but it decreases as the decay rate increases. So if a molecule that is less stable, or it gets used up more frequently, it's going to have a shorter diffusion length than a molecule that is more stable and that it's get used less and less frequently. It's important to notice that in tissues, the typical diffusion length, and this is just like a number that is kind of like a go-to. We always sort of like, it's a reference, a reference length. It's usually between 5 and 150 microns. We usually say like 100 microns. So as as to set out like a characteristic length within tissues because of, for example, availability of resources away from certain sources. Okay, so what we're gonna do is that we're gonna come back to the exercise we did with the linear decay, but instead of having linear decay, just diffusion, we're gonna add the diffusion, we're gonna add a decay. Instead of having no decay, we're gonna add decay so we can see what the concentration profile looks like, okay? And again, this takes no time, so I'll work with you. I'm gonna put out the poll, and this time I will ask people. So, so just let me like make sure that you're following through the exercise. So what we're gonna do is we're gonna create a new simulation, or we could use the previous one, right? The one with the linear gradient. Let's do that. No, what I'll do is I'll because this is quick, so I'll walk you through it just as a refresher, and also so you guys have one version of it and can continue with the exercise as you work through it. So we're gonna create a new project. Let's call it Decay, just to have some informative name. Andy, I have a question for you. If I relaunch, because my CompuCell seems to be getting slower in NanoHub, do I have any advantage by, let's say, terminating the session and relaunching it?
I'm sorry, could you say that again?
Do I have any, so for example, if my simulation is starting to get slow in NanoHub, would you recommend terminating the session and relaunching it? Or should I, or I wouldn't expect to get any advantage from it?
It's hard to say. I would say probably not.
Okay.
I mean, by that I mean if you're doing something that incurs a lot of memory leaks, then yeah, I mean you might try resetting, but that's usually not the case.
Okay, that's good to know. Right. It doesn't hurt anyone. So what I'll do is I'll try, and we can sort of like experiment and see whether that helps or not. But thank you, Andy. So we're gonna set a simulation domain that is in 100 on 100 by 100 lattice. Again, we're not introducing cells, so it doesn't matter much what we do here. We're not gonna introduce any cell type. We do want to have a field. Let's call it oxygen, or just for fun, let's call it glucose. So we change the biological molecule, and we're gonna use the diffusion solver FE. Just hit add, which I always forget to do. And we don't need cell properties and behaviors because we're not using any of those. Okay, we'll finish the simulation. And let's close the project. So if we come to the XML script, as we see, we have no cell types. We don't need them. We had what we've been playing with is a global diffusion coefficient, but we've always set the decay constant to be 0. This time we're gonna set it to be 0.001 instead. Okay. And since we don't have any cells specified, we don't have to say what happens with the cells inside cells or anything, because the secretion data, as you see here, there, since we did not add any cell type, we don't have any secretion. But just keep that in mind, because when you populate the simulation with a cell type, this gets automatically populated, and you might have cells secreting stuff even if you did not intended them to do. So make sure that you always look through the code to make sure that it's not doing anything that you didn't mean it to do. As always, let's set the value on the x-axis to be 1 at the minimum and 0 at the maximum, and periodic on the y-axis. Okay. And what we will do next is, because we want to display the, we want to be able to see the measurement of the field. Oh, I wanted to also mention this. When we do not initialize cells, Composable still initializes the uniform initializers stepable. So, but we don't have any cell type and we don't want any cells. So let's just get rid of that part. Just want to make sure that I didn't delete anything important from the XML, right? So we can get rid of the uniform initializer stepable part, and that should work. But we also want to add a plot so we can scan through the simulation domain to see what the concentration profile looks like, right? And this is important because this is the way we iterate through the domain regularly. So if people have questions in their assignments and homeworks about how to iterate through the simulation domain, please do pay attention to this part, because this is the way we usually do it. So for all plotting, we always start at the start function, and we can use the CC3D Python pull down menu, and we hit on the setup. Okay, and make sure to always edit the titles and the axes so it's informative to you and to other people. It's gonna be the concentration by location, right? The x-axis is gonna be the location that I'm gonna be measuring the concentration at, and the y-axis is gonna be the concentration. And I just want one data set, one data plot, and it's gonna be, let's call it for concentration, and then we can get rid of this part. I always prefer to do dots because it gets less noisy when it's very discontinuous. When the plot is very discontinuous, it gets less noisy. So we have set up our plot and the data that we're going to be specifying, and in the step function, all we need to do is to iterate right across the X domain, and the X domain starts at 0 and ends at self.dim.x. So for X in range 0 to self.dim.x, and this is a property that tells me basically the lattice dimension, then I'm gonna have to add what the value of the field is at that particular location. And the way I measure the value of the field at that particular location is in the CC3D Python menu. I have the chemical field manipulation. And there's two things I need. I need the value, and I'm gonna need the reference to the field. So I'm gonna start with the reference, and I can put that reference outside of my loop because it's the same reference for, it's a same reference regardless of the the location I'm looking at. And it's called oxygen. Right. So now I have grabbed that object called my field, my oxygen field, and I have this reference to it. And now not all I need is, by using this reference, I need to grab what the field concentration is. So I can use, in the 63 Python pull down menu, I get this get field value. Right. So now this snippet is going to tell me what the field concentration is at that particular location. Then I have, since I'm iterating through X, that's going to be the X point. In the Y point, it doesn't really matter, but I prefer always doing it because it's going to be uniform on the Y axis. But I always prefer to, because we set up the conditions to be periodic, but I always prefer to do it just in the middle of the simulation.
I think our field is glucose.
Oh yeah, I'm sorry. Thank you for that. Thank you for catching that up. It was going to crash on me, and I was going to spend time just trying to figure out what the problem was. Thank you. And now we need to add the plot data. And we're calling the data set, we're calling it, and instead of time, we're going to plot location and the value of the field at that location, which is value. And I mentioned last time with that the only problem with this code is this code is gonna do what we want it to do, but it's gonna overlay every step I run it. It's gonna overlay the data that I get over the data that I got the time point before. So it's gonna be just a bunch of points, just one overlaying over the other one. To avoid that from happening, since I want my plot to refresh every Monte Carlo step, I have that functionality in scientific plots, erase plot. I put it on before plotting all of the data. And then the last thing I want to do is that this is a little bit of expensive computation, so I don't have to do it every Monte Carlo step. So I'm just gonna do it every 10 Monte Carlo steps. And yep, that should work. So now that, okay, now that I've finished coding this, I'm gonna give everyone a little bit of time so they can catch up, but because I want someone to share the screen and we'll do the exercise with them. It's not gonna be complicated, but so I know at least 2 people are up to date. Who wants to share the screen? There's
You may
4
just
people
have to
now.
pick something. So you may just have to pick somebody.
Okay, everyone seems to be. So I'm gonna ask like by the, and then the next person's by the order here. It's gonna be Devin.
Okay, yeah.
Okay, I'm gonna
you have to
sharing
stop
so you can
sharing?
share.
Okay.
Yep.
Let me get this out of my way real quick. Okay.
Just do me a quick favor. Increase the dot size to 5, so we, because they're usually very small. Yeah.
Okay. I'm not sure what that error is.
Yeah, go to the XML back again.
Okay.
Just let's make sure that we call it the right name. Go. So it's just because it has a it's uppercase G upper G. So you can either modify it here or on the XML. Doesn't matter. But yeah, here it's probably easier.
Where do I need it inside the?
You need on line 26, just make that G self that field that that one. You're good.
Okay, is that the only thing then?
Yep.
Okay.
Right. So just this is fine. Show the in the cell field. Show the the glucose field, right? There you go. And then go to settings, which is the range, just to make sure that, and go to field. And this is fine. This is floating. You could also fix it to make it look better, because you know what the maximum and the minimum are. So in the range, fix the minimum to be 0 and the maximum to be 1. And hit OK. Takes 1 second. Right, and now you can play the simulation again. So, okay, someone else, like what is the difference between what we see here and we saw before with the linear gradient? What was happening in the linear gradient? I mean, this is inverted, but it's the same idea, right? In the linear gradient, the concentration was decreasing from the source, which in this case is 100, to the sink, which in this case is 0, linearly, right? There was a straight line between them. But when we added decay, then the distance, I'm sorry, the concentration profile, instead of decreasing linearly, is now decaying exponentially. And then since we started at 1, the distance that it takes to decrease by a factor of E is going to be the diffusion length that we have established, which should be also the square root of the diffusion coefficient over the decay rate. Okay, let's do something here. Devin, can you go to the model editor and plug in the steppable of the diffusion solver? Open up that one. And then, right here in the global decay coefficient, set it to the one below. Yeah, set that one to 0.
Am I allowed to set it here?
Yeah.
Doesn't seem
you
to
should,
be letting me edit.
double click on it.
Oh yeah.
0 and enter. So what's happening now?
Looks like it's going back to a linear spread.
Right, and that's because we set back the diffusion. We went back to the previous case, right, to the linear gradient, where there was just diffusion, but no decay. Okay. And set it back to, pause it for a second, and set it back to 0.01, the global decay. But now, and play it once more. I'm sorry, play it once more. Right. So you see that it goes back to the decaying function of location, but now change the global diffusion coefficient and make it 5. So what's happening now is that it's still decaying, right? But the length, the distance that takes for it to decay a factor of E is bigger now, right? Is bigger by a factor of 2, right? Because that's how much we increased it, yeah, by a factor of 2, because we increased the global diffusion coefficient by a factor of 5, by the square root of, square root of 5, yeah. So we'll continue talking about how to model different processes that alter and change the concentration of molecules in the aqueous tissue, right? This was an example of what we just saw, the difference between having a linear gradient when we have a source and a sink and just diffusion with no decay, and the exponential decay that we see when we include this decay coefficient. Now, as we keep going, there is more and more terms that we can keep adding to this diffusion equation. Some of these terms are meant to represent processes that produce and add concentration or add molecules to the field versus processes that take out some of the concentration out of that field. The processes that add concentration, we call those secretion. The processes that take up concentration, we call them uptake. And we will start with the secretion aspect. The secretion is one of the processes that cells use to release release chemicals and molecules from the inside of the cell to the outside of the cell. It's usually it is a form of active transport, which means that requires the consumption of metabolic resources. There are very complex ways of secreting, or also called exocytosis. But in general, all these mechanisms require the molecules to be packed into intracellular vesicles that are transported from the cytosol of the cell into the plasma membrane. These vesicles dock into the plasma membrane, and some lipoproteins that are in the last plasma membrane, and by docking, then the chemicals inside these vesicles get released into the extracellular environment. The net effect of this release is a change in the concentration of the molecules at the outside of the cell. This is a little bit of a more philosophical discussion that James and I were having yesterday, but from our perspective, especially in uptake, that we will talk about later, there's no real difference in the way we would implement an active transport, let's say, from the outside of the cell, from the inside to the cell to the outside, as opposed to a diffusive passive process, because all we care when we model secretion is that there is a cellular process that increases the concentration of the chemical over the domain of the cell. And we're happy to come back and talk about the details of that. So the important thing about secretion and CompuCell is that we will need to import this special plugin called Secretion Plug-In. And that is gonna give us control as to how much, at what rate, or under what conditions the molecule is secreted to the extracellular environment from the cell. We have different ways of implementing this secretion spatially. One of them is doing it at the cell boundary. What we will, if we look at this diagram here, the secretion at the cell boundary would mean that the source of the chemical is the pixels that are adjacent or that are at the border of the cell. Those are that constitute what we've been calling the surface of the cell, or the computational objects don't have like a explicit surface, but those pixels that are at the edge of the cell are the ones that are going to be secreting this field, and we call that the secretion at cell boundary. I'm going to go to this track amount. I'm going to come back to this track amount here in a second. Another option that we have is to secrete at the on the outside of the cell boundary. And this is important there is a distinction in terms of what the underlying biology is intended to read. What this distinction is intended to represent in respect to the underlying biology. But in terms of the computational implementation, in this case, what it means is that the pixels that are on the outside are adjacent to the surface pixels of the cell, or the pixels at the edge of the cell are the ones that are going to be secreting this this chemical. Two other options that we have is to have our our chemical be secreted over the cell, the whole cell, and to use that we call we use the inside cell secretion. And we also have the option of secreting at the same at the center of mass of the cell, and in that case, the chemical is released from a single pixel that is calculated to be the pixel that is at the center of mass of that cell. And we will explore in our next exercise what the differences are between these. When we load the secretion plugin in CompuCell, we get this default snippet on the CC3D ML. And the basic components of this plugin is that it's going to require us to specify what the field the cell is going to be secreting is, what the secretion type, what are the conditions under which secretion is going to happen, and we're going to have a couple of different options. Do we want our cells to secrete constantly, regardless of any other condition? We can use this secretion format where we specify what the cell type is and what the secretion rate is. So it's going to release here in this particular example, my set is releasing or secreting 0.1 units of concentration into the field per unit time, per simulation time, right? Or whether we wanted to secrete only when it's in contact with other cell types. And this is important for example, signaling processes. So the cell can start secreting a chemical only when it's in contact with some specific other type of cell. And this is something that, for example, immune cells do, right? They transduce signals as they recognize certain epitopes in the surface of target cells they are scanning for example. And lastly, we can specify that the cell will always have a uniform concentration, similarly to what we've done already with our boundary conditions, where we say that the concentration is always the same at the cells, inside the cell. And as I said, we can do we have the advantage of doing secretion in CompuCell is the advantage, you have the advantage of either specifying inside the XML with the given options that are specified in the XML and the functional forms that are available there. But you have much more flexibility because you can specify more complex functional forms for the secretion. And you can, so for example, we were building a model where cells were secreting a particular molecule called interferon, depending on the amount of interferon that they had inside, because of the activation of the interferon stimulated genes. So you can make this secretion a function of other components or other aspects of the simulation. And this gives you much more flexibility in terms of how much the concentration changes because of the cell secretion and uptake, even though we'll come back to that one later. And I want to talk about this track amount here. It is in the documentation, but it's not available in the snippets. When you do the CC3D pull down menu, you get all these options in inside cell at boundary, inside cell at boundary with contact, etc. But you don't get track amount. But you can add it, and it's supported by the current version of CompuCell. And what that would do is that not only the cell takes up, but it allows you to keep track of how much the cell is taking up. And we can do that, and how much is being secreted, for example. And we can play a little bit with that. We can have our cells die, for example, let's say they are undergoing apoptosis and they are starting to release matter, like cellular material, into the extracellular environment. We can compensate how much volume we have inside the cell with how much secretion is happening because of the cell undergoing apoptosis. We'll come back to that later. So the first exercise we'll do, though, another thing that we can do is that we can have our medium secrete. So it doesn't necessarily have to be like a explicit cell type, but we can also have our secretion, our medium secrete. And that is a particularly useful case, because then we can set up an equilibrium concentration across the tissue. And for those of you who are working on the cancer simulations, that is usually a very common way of representing metabolic availability of metabolic resources. Instead of having necessarily, let's say, resources coming from the borders inside, as we've been simulating so far, we can have our resources come from the medium, right? And then the question we ask here is, what is the equilibrium concentration if we just have medium supplying the concentration instead of, for example, the border? And we will do this ourselves. This is also a very quick simulation, but I'm gonna launch the poll because I'm gonna call them someone after we are done writing the code. I'm gonna call them someone to write it, to run it. Okay, wait, there you go. Okay, so let me stop sharing here and share my full screen. Okay, we'll start a new simulation. called, let's call it secreting medium. I promise I would terminate the session to see if that would speed it up. Let's just keep going. I'll do it. I'll do it while I keep going to the next exercise. Let's just set our usual 100 by 100. And the reason I'm doing this 100 by 100 is because we're not simulating cells. We just cared about the diffusion. And since all of this is pretty uniform, increasing the simulation domain is not adding any information. It's just making the computations longer. We're not gonna add any. We do need medium, but we don't need any other cell type. Okay, and we're gonna add, let's call it again oxygen, and please do remind me that I'm calling it this oxygen this time. And we're gonna add it with the diffusion solver, Fe. So now when we even though we don't have a explicit cell type, we still want our medium to secrete something. So we're gonna need the secretion behavior. And this part is important because once you select the secretion behavior, instead of being able to finish the closing the Widget right away, you're gonna have an additional option called Secretion Plug-In, and this will allow you to specify some of the parameters of the secretion. And what we want to do here is, just for starting, we're gonna have our medium secrete oxygen at a constant rate, and that's what we call uniform. We cannot use on contact here because medium is literally, it's always on contact with medium, so we cannot use that one. That would be called helpful when we explicitly simulate cells, but for medium, we can use either the uniform or the constant concentration. We'll play with those. And what we will do is that we will set the secretion rate of medium to be 0.2. And we add it, and then we can finish the concentration. We can finish the simulation. Okay. So I'm actually good in time. I think I might be able to actually cover everything today. We'll see. So if we come back to our XML now, yeah, the usual, we don't have any cell type, but now we have this secretion plugin here that we added, and notice that it has what we've specified so far. And the secretion rate, it's being specified inside of this plugin. And as we come down and look at our field specification, right, we notice that we have the global diffusion constant, the global decay constant. But we also see that there is also the secretion data here. And this section of the code will also define secretion data for fields when I explicitly declare cell types. So you might want to keep it in either of them, but it's preferably not on both of them. I don't have it here now. I'm not sure if it matters, but I always prefer to specify everything inside the diffusion solver step one. But right now it's specified here, so it should be fine. What we will do is that we want our boundaries to be periodic. And on the x-axis and the y-axis. And as before, even though we didn't initialize any cell type, Composite also is always going to have this uniform initializer load up. So I prefer to get rid of it just to avoid any confusions. Okay. So let's come back to something we forgot to say was the global diffusion constant and the decay constant. And what is important, this is important because if you look at the diffusion equation and we ask ourselves again, what is the concentration at equilibrium with this uniformly secreting source and uniform decay over the whole simulation domain. And if we solve that equation, the equilibrium concentration is going to be the uptake rate over the global decay rate. Or I'm sorry, no, it's not the uptake. It's the secretion. I forgot. That's a typo. It's the secretion rate over the decay rate. Let's say this decay rate to be 0.001, and given that we set our secretion rate to be 0.02 and our decay rate to be 0.01, what is the concentration, the equilibrium concentration which we would expect in our tissue? Based on this equation. Remember that it's secretion over decay. So just to clarify, what I'm saying is that if we go back and look at the diffusion equation, and under these conditions, we solve that equation, we get what the equilibrium concentration is going to be given a constant secretion rate and a uniform decay rate. And that concentration is going to be the secretion rate over the decay rate. So what is given these parameters here, what is the concentration I should expect to equilibrate at?
200.
200, right. So so let's see who's next in the list. It's gonna be Drew. Well, I mean, anyone like yeah, let's see. Is everyone following? How many people have it like up to date? I'm just literally calling down the list, but if someone has it available and wants to show it, I just want them to write the code. Okay, there's four people who got the code, who are up to date with the code. Someone wants to share it?
I can do it.
Okay, I'm gonna stop sharing. Okay, so right, but okay, so you have to comment out line 62 and 63. And lines 79 and 70.
79 and 70?
I'm sorry, 69 and 70. I'm sorry. I'm tired. 69 and 70. Right. You don't need the uniform initializer, so just delete that part, line 79 to right. And the decay constant 48 is going to be 0.001. Right, and let's just make sure that if you go up, that the decay rate is right. Okay, good. So, okay, step your simulation, open it and step the simulation. Just step it once. Good. So now go to settings and configurations, because again, we know what the equilibrium concentration is going to be. So we want to be able to see it. So go to the field settings and set your fixed minimum to be 0 and your maximum to be 200. Okay, save this. And now display the field instead of the cell field. So you see that you start basically at 0. And I want someone to explain me what's happening as you keep going with the simulation, as the simulation keeps going. Any ideas? So look at the maximum. What's the maximum? You can see the maximum of the field gets displayed here.
It's the value of the equation we just solved.
Right, which is 200, right? And if we look at the field, right, the field is uniform across the simulation domain because my field is occupied by medium everywhere, right? And since it's occupied by medium everywhere, every pixel is basically supplying chemical into the simulation, and the decay rate is constant across my simulation. It's uniform. So my concentration is uniform. My steady state or my equilibrium concentration is uniform across the tissue. And this is very important, especially Devin. This is the way in your paper and in my cancer paper, this is the way we simulate supply of resources into the simulation domain. And the underlying assumption is that our simulation domain is it has sources of metabolic resources, either oxygen or glucose, uniformly distributed across the whole tissue, unless something else happens. So that's why you get what it would be like a steady state concentration of a field or a chemical at the tissue. And this is actually something that it's experimentally measured. You could set up your parameters such that you get this concentration, equilibrium concentration, to match the experimental observations. Okay, so yeah, and the whole purpose of this was to show how this equilibrium concentration can be set and how you can have medium supply to the simulation domain. Okay, so if you don't mind, Tyler, I'm gonna take back the screen from you. I'm going a little bit faster because I want to try to cover a little as much as I can. But if you guys have further questions about this, please do interrupt me and I'll I'll come back to it. So the next exercise we're gonna do is in the previous example, we set up the medium to be the secreting the secreting cell type into a single cell be the secreting source. And then we're gonna see what the options are on Python. So for this exercise, we're gonna create a new, and again, I'm gonna issue a poll because I want people to follow along. Okay. Just 1 second, my nano hub crashed. It's very, very slow. Yeah, mine, I think crashed. Okay, what I'll do is I'll ask someone to do it. I'm gonna ask someone to pull up their screen and we'll work it together because my nano hub is not cooperating. Let's see who hasn't done it. Jeffrey, can you pull up your screen? If you don't mind.
Yeah.
Or Fambo or Drew, anyone. Okay, Jeffrey will do it. And in the meantime, I'm gonna relaunch my computer. I don't know what's happening there. Okay, so what we'll do this time is we're gonna create a simulation called single cell. And so do this the 100 by 100 and use the blob initializer because we're gonna initialize one cell in the center of the simulation domain.
Okay.
Right. And create a cell type called cell or whatever you want to call it, and freeze it for now. We can unfreeze it later. So, right. Yep. Add a chemical field, whatever you wanted to call it. Oxygen is fine. And yeah, but you go back because you have to click add, right? Do you click add?
Yeah.
No. Yeah, that happens to me all the time too. Yeah. It literally happens to me all the time. Now we want secretion.
Wait a sec, check it again. Go back. Okay.
Yeah.
You called it chemical.
Yeah, but it literally happens to me. Yeah, it literally happens to me all the time that I forget to click add. Hit continue. We want secretion. So, right. And click. And let me check what the parameters are because I found an interesting set of parameters. So set the secretion to be 1.
Still uniform.
And yeah, uniform is fine. So wait 1 second. Then I'm sorry, I put down my screen. Okay, so yeah, no, but the cell type is not gonna be medium now. It's gonna be the cell that you want to. So right, and that's fine. Launch the simulation. Okay, so yep, go to the XML and everything is fine. Look at the secretion. Now you have the cell secreting, right? It's in lines 32 to 40. You have the secretion plugin. But you also, and this is what I was talking about, you're also gonna have secretion data inside the PDE. line 60, which is a repetition of what you have in line 32 to 40. So I usually comment that out just to keep it like to have it once. Right. And we want the diffusion constant and the decay constant to be the same inside the cell and outside the cell. Okay, and set the boundary conditions to be periodic on the X and Y axis.
What did we want to do? Decay and diffusion?
It's just to be the same. Is it the same? 1, 2, 3, 4, 5. Yeah, it's the same.
Okay. And comment this out.
Right. Make those periodic. Right. So now we're going to initialize one cell in the center of the simulation. So make it right. Make it width 5 and radius 3. And yeah, now launch your simulation. And now, yeah, you can display the chemical field. Right. Again, there's always a problem of how the field is displayed. If you go to the configurations, you might want to figure out whether it's fixed or not. Right. So it's fixed. So unfixed it because we fixed it in the previous simulation. So those are saved. And now you can see that our cell is a source of the chemical into the field, right? And in Jeffrey's case, he's updating the screen every 100 Monte Carlo step. That's why I think it looks like it's going slower, but it's in reality it's just going as fast as in everyone's other simulations. Okay, so what we want to do now, and Geoffrey, I'm sorry, but I'm gonna keep using you as an example.
Okay.
Let's make the cell bigger to see how the secretion is happening inside the cell.
Okay. Do you want me to? Can you do that?
Just go to the, yeah, just go to. Don't close. You don't have to close this, but it's fine. But yeah, go here. Line 90, increase the width of the cell to 10 and set the radius to 5 and relaunch the simulation. And I wanted to plot the field because that's what's interesting. But I think we'll do that in the homework. So if you zoom in into the cell, can you guess how the secretion is happening by how the cell profile looks like? Because you see the boundary of the cell is the yellow boundary, right?
Mm-hmm.
So my guess is that secretion is happening at the center of mass of the cell, right? Because it's not uniform inside the cell. And also it doesn't look like it's secreting at the border either. Right, it's hard to see the cell because it's in the yellow. But if you run it again from the beginning, you'll see it more. Just step it. Right, you can see it there, right there. Yeah. So when you set it in the XML, the secretion is happening inside the at the center of mass of the cell. So I don't want to go super quick, but I do want you guys to see the syntax, even if we don't get to uptake. So what we'll do, Geoffrey, is now what we want to do is to have a little bit of more control over how secretion is happening. You guys see that you can specify a constant rate here. And you have other options. Let's explore them. Go back to the XML. And in the secretion plugin part, you see that you have secretion, but you could also have constant value. It's just not displayed here. Go down into the diffusion solver to the PDE solver. See, you can see, yeah, you see the options there. Line 62.
Oh yeah.
Copy that line and paste it in line 38. So now instead of secreting at a constant rate, what we'll do is we'll have our cell have a constant value. Same way we've been setting the value of the border to be constant. So the cell will behave just like the border was behaving in our previous simulations. Yep, open it in Player. So what's the difference now? Look at the maximum value of the field, right? It's almost 0.01, which is what you set the constant concentration to be.
Mm-hmm.
What happens also is that the cell, every pixel of the cell, if you zoom in into the cell, every pixel of the cell has the same concentration, right? Which is 0.01, the one you set it to be, right?
Mm-hmm. Yeah.
You see that? Did everyone else see it? Let me see what the poll is saying. Yeah, people are following along. Okay, so yeah, before we go, I don't want to go too quickly, but before we go, I want to show you guys how to have more control of secretion using the Python file. So let's go back to Twitter. So the first thing is, if you want to have control over the Python, you need anything that is specified in the XML is going to override anything you specify on Python. So if you want to be able to specify things in the XML, and we saw this with the volume, you have to comment them out in the XML. So go back to the XML and comment out the secretion. Leave the plugin, but comment out the specification, right? You can just delete it. So here you will get an error because in XML, for some reason, you cannot have comments over comments. So lines 36 to 34, those will throw an error. So you can just delete it. You need the plugin, but you cannot specify any information inside of it. Okay, good. Right, there you go. So now if we go to the steppable, let's think for a second. I know I'm speeding up a little bit, but let's think for a second. What we want is we want to secrete inside. We want our cell to secrete, right? So what we need is to iterate over our cells, right? In this case, we just have one, but we could have plenty of cells doing the same thing. And we want it to secrete. So inside the for cell loop, right, we're going to have some secretion happening right there in line 22. But we don't know the syntax. So what would you do if you don't know the syntax for something?
Go up here to
right. And there is an actual secretion uptake option down here.
Yeah. I see here, yeah.
Yeah. And then you have all these options, right? Inside, secrete inside cell, secrete the ones I was just explaining: secrete inside cell, secrete inside cell at boundary, secrete inside cell at center of mass. We saw that the implementation in XML was secreting at the center of mass. And we also play with this secrete inside cell constant concentration. So let's just to try something else, let's secrete cell at boundary and see what the syntax is for that one. Okay, so you get this snippet code. It has a little bit of some instructions. It's asking you to make sure that the secretion plugin is loaded. We did that already. We asking you to make sure that the field is defined in the PDE solvers. We have a field called chemical in the specified. And it's saying that this secretor object that we're defining in line 25, you can use it for different cells. It's not a secretor per cell. So you can take that part of the code and define it outside of the loop, of the 4-cell loop. Right. So now just make sure that the field name corresponds to the one that you have, and let's make it 1 here. Just so it's gonna secrete 1 unit at the boundary, per pixel at the boundary. Okay, now go back to your CompuCell simulation and run this one. Okay, so if you just step 1 second and before running the whole simulation. So what do you guys see there? What is secreting? At boundary? Is it inside or outside? I think it's inside of the boundary, right?
It's going both ways, I think.
Yeah, but because the yellow line, it specifies the cell boundary, right? And you can see that the pixels inside of that are the ones that are releasing inside cell that boundary, right? So you see that the concentration inside of the cell, that the field is just always secreting at the boundary of the cell.
Yeah.
And it's gonna secrete whatever rate you specify per pixel, right? So it's every pixel is gonna always release 1 unit. Okay, go back to the step about to the tweeted, and let's try a different one instead of secreting at cell boundary. How do I delete my arrow from there?
You turn off the annotation.
Yeah. I feel like one of those professors. How do I turn on the projector? So now secrete
Said also
outside cell, right, at boundary. And we have all of that data, so all we need is, right, you already have it, right? You already have it in 20 because that secretor is the same for every operation. And set it again to 1. And then let's see how it looks like. So, right, pause it right there. So you can see that the yellow line is the border of the cell, and if at the first step, relaunch the first step, you see what is secreting now. Secreting on the pixels that are outside, on the other side of the edge of the cell. And you could make this secretion a very complicated function of whatever. Let's say you want your secretion to increase linearly with the amount of time. So you could come back to Python, your Python file, and at the secretion rate where you have 1, make it 1 times MCS, which refers to my secretion time, and run that now. So by the way, you don't have to, and I've seen other people do like this as well. You don't have to close Compusol and relaunch it every time. Like you update the code and it automatically updates the code on the. So what's happening now is, and look at the maximum, how big it is now. It's that my secretion rate is increasing as time goes on. And this is just one example of how much control you get by using this Python step out, because then your secretion can be as complicated a function as you want. So for example, going back to what Tyler was talking earlier today, that he has this intracellular model of signaling pathway. You can have then the secretion rate, the secretion function, make it the secretion rate, make it a function of whatever is happening inside the cell. And if you guys ever want to look at our interferon model, the interferon model secretes interferon as a function of whatever, how much of the interferon is being induced inside the cell. Okay. So what I'm gonna do is I'm gonna take back the screen really quick. In the last 8 minutes, I'm gonna walk really quick over the idea of uptake, even though I'm not gonna be able to implement it right now. Uptake, secretion is when cell releases something to the extracellular environment. Uptake refers to the opposite process, and they're where cells take up stuff from the extracellular environment. Endocytosis usually refers to that process of taking up. It usually involves to some sort of energy expense from the cell. Although, as I mentioned before, chemicals can diffuse into the cells due to gradients across the membrane, and those will also be represented computationally by uptaking out of the available concentration field. What is important to notice about mathematically representing uptake is that the concentration in the extracellular environment should never go negative. Because the cell is always going to uptake based on whatever is out there. So the uptake rate has to be a function of the available concentration in the outside of the cell. And another important thing to notice is that if you plot this, depending on the ratio of the diffusion and the uptake, if you plot the profile of the field, what you would notice is that the field remains relatively stable, and then it decreases rapidly as it approaches the plasma membrane where the cell is uptaking. But the specific profile of the concentration looks like around the cell is gonna, of course, depend on the uptake rate and also the diffusion constant. But what is important to keep in mind is that uptake will always have to be a function of whatever is available out there. Compusol has its own uptake function that is predefined. And the idea of this function, it's a little bit complicated, but once you start playing with it, you sort of get some familiarity with it. Is that the uptake is always, as I said, the uptake is always going to be a function of the concentration. And there are two parameters that we can specify. we can specify a maximum uptake and a relative uptake. And the difference is that maximum uptake is the amount that is going to be taken when the concentration is above a particular threshold. So when the concentration is above the maximum uptake, the cell basically is going to take the maximum uptake. But when the concentration in the outside of the cell is below the max uptake, or if the concentration is below the max uptake, then the cell takes a fraction of what is out there. And the idea then is that this function gives you two different functionalities. When the concentration is above a certain threshold, the cell behaves pretty much like a sink, where it takes everything that is out, well, it takes at a constant rate. But when the concentration is below center threshold, then the cell is gonna take at a fraction of what is available out there. And this is to represent two things, right? To represent the first thing that this functional form represents is that there is a maximum rate at which cells can take up stuff from the environment. Cells, let's say you have a huge concentration of glucose, no matter how much, if you keep adding glucose into the environment, cells are not gonna start taking faster because you're just adding more glucose. There is a limit there's a limit at how fast cells can uptake glucose that is gonna be determined intracellularly by how much glucose the cell is consuming. But also, if the concentration falls below certain threshold, or as the concentration decreases, the cell diverges away from this maximum uptakes and just starts taking a fraction of whatever is available out there, depending on the efficiency of the cell uptake. And that is what this function is intended to represent. The uptake has to be specified in the step-out function, and it's very similar to what we just did with the secretion. We have to load our secretor, where we specify what is the field that is going to be uptaken, and then inside the cell loop, we use this uptake function that has the same abilities. It has uptake inside cell and uptake at the cell boundary. And then we have, again, to specify the two parameters, the maximum uptake rate and the relative uptake rate. So here is an example of what that code would look like inside when you put it inside the cell loop. And I'm gonna leave this as an homework as well. In the homework, what you will do is exactly what we just did right now, where we created a frozen cell, but instead of having the cell secrete, we're gonna have the medium supply and the cell take up the whatever is being supplied by medium. And what's interesting about this, and I'll show you just as a teaser, is that you could, by combining how much the cell is taking and how much is available to the cell, you can start determining, for example, changes in cell types, or changing in the type of the cell due to, for example, how much stuff is taken or how much stuff is still in the extracellular environment. We usually do that a lot with exposure, for example, to virus in the extracellular environment. That helps determine when the cells become infected. And ultimately, you could combine this uptake function with a growth function, in which instead of the growth, as we did before, the growth being determined by how much space the cell has available, how much pressure the cell is experiencing. You can make the growth rate a function of how much nutrients the cell is uptaking from the environment. And I'm gonna add some teaser homework questions about this, but just feel free to reach out if you have come across any questions. So it's time now. Thank you all, and thank you for participating. I think that's helpful for all of us. So we'll probably keep doing that in the future. And I'll see you guys next week.